Good morning, students. Today we are going to discuss hypersensitive reactions. Okay, so here we will see how many types of hypersensitive reactions are there, then how a normal immune response and a hypersensitive response is different. Okay, so let us see. Inappropriate immune response, which can have deleterious effects resulting in significant tissue damage or even death, is termed as hypersensitivity or allergy. Gell and Coombs classification of hypersensitivity is most widely used, and they have classified hypersensitivity into four classes: type 1, type 2, type 3, and type 4. So, we are going to see each of these types in detail. In type 1 hypersensitivity, which is also called as immediate hypersensitivity, an allergic reaction is provoked by re-exposure to a specific type of antigen referred to as an allergen. So you might have heard that some people are allergic to pollen, dust, or even sometimes gluten, one of the protein in wheat. So these are all allergens which can initiate type 1 hypersensitivity on their re-exposure. Exposure may be by ingestion, inhalation, injection, or direct contact. So, any of these can lead to the hypersensitivity reaction. In type 1 hypersensitivity, B cells are stimulated by CD4 positive T helper cell 2 to produce IgE antibodies specific to antigen. So this is to be noted here that in type 1 hypersensitivity, IgE antibodies specific to the antigen are produced. Okay. So the difference between a normal infectious immune response and a type 1 hypersensitivity response is that the antibody is IgE in case of type 1 hypersensitivity, whereas in other reactions, other cases, usually other immunoglobulins are produced. Then type 1 hypersensitive response is very fast and it occurs within minutes rather than multiple hours or days. Okay. And free antigens cross-link the IgE on mast cells and basophils, which causes release of vasoactive molecules. Okay, so we are going to see how it causes the cross reactivity and all those things. All right. Here in this case, this class of antibody, that is IgE, binds with high affinity Fc receptor on the surface of tissue mast cells and blood basophils. So in this picture you can see this is mast cell and blood basophil cells, the brown color one, and IgE antibody. The Fc receptor of IgE is binding on the surface of these cells. So you see the receptors, the pink color is the pink color on the surface of these brown colored cells are the receptors for Fc receptor of IgE. Okay. Mast cell and basophils coated by IgE are said to be sensitized. So as soon as these antibodies IgE they bind on the surface of mast cells and basophils, they are called sensitized. A later exposure to same allergen cross-links the membrane-bound IgE on sensitized mast cell and basophils, causing degranulation of these cells. So you see the green color one, that is allergen. So when the re-exposure of these allergen happens, these sensitized cells, they cross-link with each other. So you can see the two antibodies are their FAB side of the antibody is binding to the allergen, that is green in color, and then two antibodies are reacting with same allergen. This is called cross-linking, which in turn results in degranulation of these cells. Typical manifestation includes systemic anaphylaxis and localized anaphylaxis such as hay fever, asthma, hive, food allergy, and eczema. Systemic anaphylaxis is the one where the infection, the hypersensitive reaction, it it spread from one organ to other organ. It is not localized anymore. Whereas in case of localized, it is just it remains to one place. In case of type 2 hypersensitive reaction, an antibody mediated process where IgG, IgM antibodies are directed against antigens on cells, such as circulating red blood cells. Here I will show you with an example that if a transfusion happens with wrong blood type, then how it leads to type 2 hypersensitive reaction. Okay. It could also happen with extracellular material such as basement of membrane. This subsequently leads to cell lysis, tissue damage, or loss of function through mechanisms such as complement activation via classical complement pathway. We have discussed complement activation and all these three different pathways: classical, alternate, and lectin. So here we are going to see how complement activation happens via classical complement pathway. Antibody dependent cell mediated cytotoxicity. It can also lead to ADCC. I'm sure you all remember we have discussed ADCC in way more detail in our unit 2. So if you want to refer, you can go back and see, because here we are going to just briefly overview that how these type of reactions, that is type 2 hypersensitive reaction, can lead to complement activation and ADCC. Here in this case, antibody directed against cell surface antigen mediates cell destruction via complement activation and ADCC. For example, complement dependent type 2 hypersensitivity is an acute hemolytic transfusion reaction following transfusion of ABO of incompatible blood. Okay. So what happens is preformed antibody, predominantly IgM, against donor cells antigen not found in an individual of particular blood group. For example, anti-A IgM in an individual with blood group B, okay, binds to donor cell surface and leads to rapid complement mediated hemolysis and potentially life-threatening clinical consequences. So you can see in this picture the brown cells are the target cell, or you can say donor cells. These are the blood cells of donor which are incompatible with the blood group of host. So what happens is IgM anti-A, these antibodies, they bind to the antigen and they can lead to ADCC, because this is cytotoxic cell. They lead to ADCC, and that way they cause lysis of the cell, and also they can initiate a complement activation. Okay. In type 3 hypersensitive reaction, it is also called as immune complex mediated hypersensitivity. Antigen antibody complexes deposit in various tissues, induce complement activation and an ensuing inflammatory response mediated by massive infiltration of neutrophils. So what happens is large complexes can be cleared by macrophages very easily. Okay, they can be identified and removed from the site very easily. But the small immune complexes cannot be identified very easily by the macrophages. So these small immune complexes insert themselves into small blood vessels, joints, and glomeruli, causing symptoms. Unlike the free variant, a small immune complex bound to site of deposition like blood vessel wall are far more capable of interacting with complement. So these small immune complexes, they can activate inflammatory response, which is mediated by massive infiltration of neutrophils and which causes type 3 hypersensitive reactions. In case of type 4 hypersensitive reactions, often also called as DTH, delayed type hypersensitivity, because previously you have seen all the types 1 through 3, they are all they give reaction in small time span immediately, but this one is delayed type of hypersensitivity, as the reaction takes several days to develop. Unlike the other types, it is not antibody mediated, but it is rather cell mediated response. Sensitized T helper cell 1 cell releases cytokines that activates macrophages or T cytotoxic cells, which mediate direct cellular damage. So here you see the blue cells, okay? So as soon as these are helper cell, okay, and then these are called sensitized as soon as the antigen binds to them. So once they become sensitized, they release cytokines. The overreaction of helper T cells and overproduction of cytokines damages tissues, causes inflammation and cell death. This is how type 4 hypersensitive reactions happens. That's all for today. Tomorrow we are going to discuss inflammation. Till then, take care, keep studying, okay? Take good care of yourself and your health, okay? Bye.